Welcome to Học Tiếng Anh với Joseph, where we build vocabulary and practice pronunciation through reading. Continuing with The Cat in the Box, A History of Science in 100 Experiments, we will read story number 64. Chúng ta sẽ đọc câu chuyện số 64, The Discovery of Nucleic Acids. Some discoveries are hard to pin down to one experiment or one person. The discovery of the life molecules RNA and DNA involved many people and many experiments over nearly 100 years. But the person who actually gave these molecules their names was Phoebus Levine, a Russian-born American working at the Rockefeller Institute of Medical Research. The story really begins, however, with experiments carried out by a Swiss biochemist, Friedrich Michel, working at the University of Tübingen in the 1860s. By that time, it was known that proteins are the most important physical substances in the body, and Michel wanted to identify the proteins involved in the chemistry of the cell, a key to the workings of life. He got his raw material from bath-soaked bandages obtained from a nearby surgical clinic and isolated the white blood cells known as leukocytes from the pus. Living cells are like little bags of watery jelly called cytoplasm, with a more compact central nucleus, the term Rutherford later borrowed to describe atomic structure. Michel found that the watery jelly is indeed rich in proteins, but he was also able to remove this outer material and collect enough nuclei to analyze them separately. He found that the nuclei were not made of protein but of a different material, which he dubbed nuclein. Just like other molecules of life, it contained a lot of carbon, hydrogen, oxygen, and nitrogen, but it also contained phosphorus, which is not found in any protein. He wrote, "I think that the given analyses, as incomplete as they might be, show that we are not working with some random mixture, but with a chemical individual or a mixture of very closely related entities." But he was not able to work out the structure of the large nuclein protein molecules. In later work, he found that these molecules contain several acidic groups, and the term nucleic acid began to be used to describe nuclein by the end of the 1880s. In the following years, analysis of nucleic acids showed that there are two types. One contains 4 subunits known as bases, with the names adenine, guanine, cytosine, and thymine, often referred by their initials as A, G, C, and T. The other nucleic acid contains a different base, uracil, instead of thymine. This was the state of play in the second half of the first decade of the 20th century, when Phoebus Levine began experimenting with nucleic acid derived from yeast cells. This contained about equal amounts of A, G, C, and U, plus phosphate groups, and a carbohydrate group that had not been identified. In 1909, he isolated and identified this as a sugar group, ribose. The sugars themselves are each built around carbon rings similar to the ones Kekule described, but with 4 carbon atoms and 1 oxygen atom linked, forming pentagons instead of hexagons. Levine showed that the components of the nucleic acid are themselves linked in units each made up of one phosphate, one sugar, and one base, and he called these units nucleotides. Nobody knew, however, how the components of the nucleic acids were joined together. Levine's idea was that the nucleic acid molecule was made up of a string of these nucleotides joined together like a vertebrae of your spine, and in 1909 he gave the name ribonucleic acid to the molecule. As there were 4 bases present in the equal numbers in what became known as RNA, he suggested that each molecule was made up of a short chain of 4 nucleotides, one with each of the 4 bases. This became known as the tetranucleotide hypothesis, which held sway for decades. One consequence of this was that it reinforced the prevailing idea that the really important molecules of life are all proteins, and that the nucleic acid simply provided some sort of scaffolding to which protein molecules were attached. It wasn't until 1929 that Levine discovered that the nucleic acid derived from thymus cells contained a different sugar group as well as having a T instead of U, because each molecule of this sugar group has one oxygen molecule less than the corresponding ribose group. He called it deoxyribose, and the nucleic acid became known as deoxyribonucleic acid. The name of the 2 nucleic acids are often shortened slightly to ribonucleic acid, RNA, and deoxyribonucleic acid, DNA. Levine still thought that the nucleotides in a DNA molecules are always linked in the same order, perhaps ACTG, 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 and so on. But the first clue that there is something more to DNA than mere scaffolding had already emerged in 1928, the year before the DNA was named. Moving on to the vocabulary section. Proteins, proteins, soaked bandages, leukocytes, cytoplasm or ketoplasm, not too sure, forgot. Nucleus, nuclei, nuclein, nucleic acid. Many nuclei, that's the root. Nucleus, nuclei, nuclein, nucleic acids. Pentagons, hexagons. The pentagon, hexagon. Nucleotides. Vertebrae, ribonucleic acid, ribose nucleic acid, scaffolding. Thank you very much for your time, and I will see you in the next story. Bye for now.